Okay. Hi, yeah, I'm Josh. I'm a graduate student with Mark Kasevich here at Stanford. I'll be telling you about the work we've been doing on cavity-enhanced microscopy, particularly for biosensing applications. Um, I should also mention that Mark's group has a cavity QED experiment for strontium atom interferometry. I wish I could tell you more about that, but you'd be much better served by talking to Guillermo, Eric, and Rose, the other graduate students on that project. So the main challenge for optical microscopy in the biosciences is that amino acids, proteins, cells, the things that you and I are made of, just don't interact very strongly with light. Not only that, we have to do our measurements in these environments that are warm, wet, and often alive. And so what that means is that being quantitative, precise, and accurate is very challenging. These samples are dynamic on a large range of timescales. They're heterogeneous. They're noisy, both metrologically and biologically. And importantly, the optics that I have to use are quite lossy. Nevertheless, fluorescence microscopy and innovations in chemistry and biology have been successful in teaching us about the structure of living things. So much so that when artists like David Goodsill here draw cross sections of things like E. coli cells, we have sufficient knowledge to do this with molecular level detail. You can see here in orange DNA, there's purple ribosomes, green lipid membranes. And these images are just so detailed now that scientists actually think it's worth storing these things on the protein data bank, where spectroscopists who take protein crystal structures with X-rays at nanometer scale resolutions also go ahead and upload their data. So these images and the structural information is mostly generated through fluorescence microscopy. So how does this technique work? I pump a chromophore with light, and it'll absorb and go into this excited electronic state, A. Typically, it'll quickly relax in this electronic excited state manifold non-radiatively, and then emit a photon to go back into the electronic ground state. So this photon will be redshifted, right? And in both of these states, I basically have a continuum of modes because I have this very bulky molecule that can vibrate. And so I get these very broad absorption and emission lines that are shifted somewhat. And in fluorescence, basically what you do is you put in a spectral filter between where you're exciting your fluorophore and where it's emitting to isolate the emission and ideally do a totally background-free measurement. So this relatively simple idea has led to advances in single molecule spectroscopy, super resolved imaging, deep brain imaging, optogenetics, and generally a really deep level of understanding and control over the biochemistry and the photophysics of these molecular dyes and fluorescent proteins. So much so now that people are even talking about genetically encoded spin qubits that will also fluoresce. And so if you want to hear more about that, you should talk to Sean Bird. But this fluorescence technique also has limitations. So for one thing, most biomolecules do not naturally fluoresce. Randy mentioned that some of them do in the UV, but as we generally know, standing under a UV lamp is not good for your health. Right? Instead, what we have to do is we attach these molecular labels or these genetically encodable fluorophores to get some idea of what's going on. But these things are big and bulky, so they can perturb the structure or the biology that I'm trying to study. I'm also putting real population in this electronic excited state. And so these molecules can undergo deleterious reactions with other things in solution. And so they go dark after a while. They undergo what's so-called bleaching. I can also have other excited state processes like singlet to triplet interconversions, which change the fluorescence intensity that I'll see as a function of time. And then for deep tissue imaging, I'm limited by scatter. And if for resolution, I'm limited basically by the diffraction limit of light. And spectrally, of course, these emission bandwidths are very, very broad. So I'm not being very efficient if I want to do something like multiplex sensing where I look at many different species. So an emerging technique to this is stimulated Raman scattering microscopy, where we're going to generate contrast from the intrinsic molecular vibrational modes. So to understand what's going on here, let's just think about spontaneous Raman scattering. If I send in light to some molecule far detuned from resonance, I can get spontaneous scattering events where the photon loses some energy and it excites a molecular bond. So that bond starts vibrating, right? And I get out redshifted photons. And if I disperse those on a spectrometer, I can actually map out the vibrations of the molecule that I'm looking at. So this is dimethyl sulfoxide. It's just a common organic solvent. And there's basically three regions that you should understand in this vibrational spectrum. At high wave number, 3,000 inverse centimeters or so, we get what's called the CH stretch region. Basically, every biomolecule will vibrate here. And the two peaks you're seeing are the symmetric and the anti-symmetric carbon-hydrogen stretching vibrations. In the low energy regime, below about 1,800 nanometers, wave number, excuse me, we have the fingerprint region. So called this because different vibrational groups will vibrate here with characteristic frequencies. So you can see the carbon-sulfur symmetric and asymmetric stretches, as well as this sulfur-oxygen stretch. And in the middle, there's also this region between 1800 and 2800 wave number, where basically nothing that your body makes actually vibrates. And so this is very useful for labeling applications for Raman, but we're not going to talk about that too much. The important thing is that these vibrational lines are intrinsic to the molecule and they're characteristic. So if I can detect them, I don't have to rely on some kind of external label that can perturb the function that I'm trying to see. But they're also particularly narrow in energy. So they're very useful from a sensing perspective. In this case, we can also look at a single line move around, for example, in which case, in some sense, I'm doing a bond selective measurement that might be sensitive to something like a local electric field, either that I apply or that comes from the solvent that the thing I'm studying is dissolved in. But this technique is very weak. When I pump these molecules, I basically get emission into 4 pi and from all of the different vibrations. So what I might like to do is increase that scattering rate and choose a particular bond to target. So we do this by just driving these stimulated 2-photon transitions from the ground electronic state to the excited vibrational state, all in the ground electronic manifold, by sending in a lower energy Stokes beam, lower frequency beam. And what happens now is that a photon from the high energy pump in blue will be absorbed. A photon will be stimulated into the lower energy Stokes beam simultaneously with the excitement of a molecular vibrational mode. So this technique still requires very, very high intensities, so high, in fact, that I usually cook most of the cells that I'm trying to look at in the microscope. And so a clever physicist's idea for how to get around this problem would be, okay, I need to increase my signal to noise per dose. Let's use squeezed light. And Warwick Bowen's group in Australia has been really successful in this regard. They've demonstrated a state-of-the-art SRS microscope using squeezed light. So they do imaging of polystyrene beads, but also these yeast cells with effective squeezings on the order of 1.3 dB, signal to noise enhancement. And this comes from the suppressed fluctuations in the optical field due to the squeezing. Now, this being a cavity workshop, of course, I'm going to tell you about how we use cavities to do this enhanced measurements, right? But what we want is a formal way to compare these various techniques. So we're going to just talk through a Fisher information calculation. This won't be too technical. If you're not familiar with this kind of formalism, just think of the information as being proportional to 1 over the variance in my measurement. And so for stimulated Raman, we're measuring a small intensity change, either due to absorption or gain. And we're going to compare to the absolute quantum limit, normalized to the amount of dose that I've applied to my sample. So it's known for absorption estimation that Fock state probes are going to be quantum optimal measurement devices. So those, for all transmissions through my sample on the x-axis here, will give me an information of 1. That's the quantum limit, right, per the number of photons that I send in. And if I instead do a classical measurement in what we call single pass, as in a normal microscope, I get this curve that goes like 1 minus eta, or equivalently, my quantum enhancement is 1 over 1 minus eta, where eta is the transmission through the sample. And this is referenced in the title of the session, of course, but you can do better. So for example, if I squeeze the light that I send into my sample, I can get an increase in information per dose due to the reduction of the fluctuations in the optical field, which we're assuming are due to shot noise for the single pass case, right? Now, alternatively, what we found is that both for absorption estimation and for phase estimation, if you tune up your cavity correctly, you get a quantum optimal measurement. And so in red here at the top, again, you can see that cavities can saturate the quantum limit on information using only classical light over the whole entire range of transmissions. But of course, I have to pick the particular parameters of my cavity. And so this actually will limit the range I'm interested in. Nevertheless, we're going to work at a transmission of about 0.9. This is mostly limited by the microscope objectives that we use in our sample. And so if we choose the in and out coupling of the cavity correctly, we can get enhancements that come within just a few percent of the absolute quantum limit per dose. So how do we build a microscope like this? Well, we're going to start with just our microscope objectives, and we'll just enclose them in an optical cavity. So this is a 3.75 meter long cavity. It has a free spectral range of 80 megahertz. This is chosen to match the repetition rate of our dual output femtosecond laser. And I already told you that the loss per sample interaction is on the order of 10% or so. So to get a quantum optimal measurement, you want to make your input coupling as small as possible and make your output coupler matched to the round trip loss. We're essentially impedance matching to the output coupler here to be the most sensitive. Now, the reason we had some difference between the absolute quantum limit and the peak of our cavity's information curve, of course, is that the input coupling can't actually be zero. So I lose a little bit of information there. So what this gives you is a finesse of 30. Now, unfortunately, this isn't the worst finesse that we've seen today, but it's pretty close. And so in the frequency domain, what you get is these very broad cavity lines that are perfectly overlapped and locked to the teeth of the laser, equally spaced by 80 megahertz. We lock the whole thing with PDH, so we imprint phase sidebands on the Stokes, the red Stokes with an EOM. And then to do the Raman measurement, we amplitude modulate the pump, and we detect with lock-in at the output the amplitude modulation that's transferred to the Stokes by this stimulated Raman process. And so this will just directly be proportional to the Stokes power and the pump power times the number of molecules in the focal volume. And how big is that focal volume? Well, we get cavity wastes of about 1.4 microns between these two objectives. If you think about a closed transition here, then that's a single atom cooperativity of about 0.8. But of course, we have a liquid sample here. It's a condensed phase. So for these spectroscopy experiments we actually have 10 to the 11 molecules in that focal volume. In a biological cell, the protein concentration is lower, of course. It's about 10 to the 7 molecules per focal volume. But those are the regimes that we're working in. Now, how do we do a sensitive spectroscopic measurement where I want spectral resolution on the order of 10 wave number or so? That's about a millielectron volt, right? But use a femtosecond pulse. We use this technique called spectral focusing. So what I'm going to do is I'm going to chirp both of my pulses in time. And if I chirp them by the same amount at any instantaneous moment, there's a fixed energy difference between the frequencies that are present in the sample. And if I match this energy difference to the Raman line, right, I'll basically drive the transition with all of the light that's present. And if I just time delay the two pulses relative to one another, I change that energy difference. So by sweeping a delay stage, I can just go ahead and collect my whole Raman spectrum by tuning over all of the possible energy differences. So we validate the performance of this instrument in spectroscopy. So here's our molecule DMSO again, right? And we're focusing on that carbon-hydrogen region, that carbon-hydrogen stretching region. You can see here the symmetric and the anti-symmetric modes. And then as you'd expect for a shot noise limited measurement, as we just increase the dose that the sample is seeing, right? My signal goes up linearly, and so too does the noise power. We then make a comparison with a single pass measurement. Basically, that's what we're normalizing to on the left here. If you do these cavity enhanced measurements, we get SNR enhancements up to about 8 dB, limited by technical noise from a photodiode. So this corresponds to an effective squeezing at the output measurement of about 9 dB in sensitivity for spectroscopy, with the ability to tune between these various Raman modes and thus also gain spectroscopic information. In imaging, we get slightly lower signal to noise improvements. This is due to additional loss in these biological samples. So what you see here are some images of HeLa cells. These are living. We put them right in the microscope at the cavity waist, and then we scan the sample through that waist to generate these images. So you can see a range of cellular morphologies here. It's a little blurred out by the big projector, right? But you see these long cellular projectors, these bulky nuclei, and there's also additional subcellular structures that are scattering more or less Raman light. So kind of zoomed out on top, a little more zoomed in on the bottom. But of course, I told you that the interesting thing about Raman spectroscopy is that we also get this spectroscopic information. And so this carbon-hydrogen stretch regime that we're probing is very busy for biomolecules, particularly in cells. But there still are characteristic features that correspond to different macromolecules. So a lot of work has gone into understanding just how, for example, the abundance of carbon-carbon double bonds in something like a lipid shifts the vibrational resonance of a carbon-hydrogen stretch. And so people have mapped out where I should look if I primarily want to see lipids or proteins or things, their DNA. And so what we can do is just take images with our delay stage set at different positions, right? And so this kind of acts like a mapping of the concentration of different biomolecules in our cells. So this peak around 2850 wave number, that corresponds to lipids primarily. This peak at 2912, that's where we see the protein channel. And at 2967, this would be DNA. And we combine all of these things into a hyperspectral image, right? And what you see immediately is the DNA lights up right in the nuclei of the cells. It's surrounded by this big blob of protein, right? And then on the out of the edges, I start to get more yellow or red colors, basically as my lipid membrane starts to take up more of my focal volume. So this is micron scale chemical imaging in living cells. We have spectroscopic resolution on the order of 10 wave number, right, for doing molecular identification and vibrational tagging. And these enhancements are just far outperforming kind of the near-term prospects for squeeze light sensitivity in these kind of Raman microscopes that can take cell images on the order of about a minute or so. So we think these cavities are really promising for enhancing the sensitivity in bioimaging in this regard. The dream of this field, of course, would be to have single molecule sensitivity, where I've tuned up my cavity sufficiently that when one macromolecule diffuses into the focal volume, just like Randy was showing, right, I get a click on my detector. Or maybe even more interestingly, I'm tuned up so that when I get a click on my detector, not only do I know that there's a molecule there, but I know that I put it in a vibrationally excited state, the one that I'm stimulating, for example. So we're really excited about this. But you might ask a follow-up question to these microscopy experiments. Okay, you've done imaging with fermions, bosons, got ahead of myself. Why not do this with fermions? Maybe more interestingly, what if I want to look at something smaller, right? So this is a nuclear pore complex. There's about 200 to 2,000 of these in every one of your cells. They basically regulate the transport of molecules in and away from your DNA. Now, this channel is only about 50 nanometers wide. It's already far below the diffraction limit of light, right? And it's made up of about 30 unique proteins, of which there are copies of a few. But these days, we can actually reconstruct this complex and see all of those constituent parts with atomic scale resolution. How do we do this? We use the much shorter wavelengths of electrons to do high resolution imaging. Now, if you're a biomolecule, an electron coming in at 80% of the speed of light, which is pretty typical, that's bad news, right? So these electrons with doses of just a few electrons per square angstrom basically break every chemical bond in something like a protein. And so what we actually have to do is freeze the sample to lock those nuclei in place. And this allows us to get just enough signal to noise that I can orient many copies of that molecule in space and then average the heck out of it. So for an image like this, the poor PhD students had to collect 617 what are called tilt series. This is you expose your sample to a range of electron incident angles, right? They looked at over 10,000 individual what are called single particles, and then you plug it into an extensive data analysis and machine learning pipeline. At the end of that, it spits out this beautiful image, right, of this single protein complex that makes up a tiny fraction, of your cell. But the point is, what are we missing here? We're missing conformations. We've done a lot of averaging, and so we can't see those kind of stranger protein orientations that would be totally neglected by this algorithm. We're, of course, missing out on dynamics, right? We've frozen our sample, and so we're looking at kind of all of the averages again. And there's still a set of proteins that we can't see because their masses are just too low to get sufficient contrast to do this image overlaying. So what we want to do again is increase the amount of information available from our measurement at the per particle sample interaction level. Now in high resolution TEM, I basically get no amplitude contrast from a biomolecule. What I have is a phase estimation problem. My molecule adds a little bit of phase to the electron beam, or equivalently, I can think about my sample as being a Fourier sum over just a set of diffraction gratings. And it's the diffraction from the sample that adds a phase to one of the electrons, and those different beams interfere in the far field to give me my image. So, of course, for quantum states, right, again, we have a quantum limit that can be asymptotically approached by squeezing. And this is kind of independent of boson or fermionic statistics. Now, for a while, the field was excited about using Newton states for phase estimation and interferometry. These are, of course, very sensitive to losses, so they do get outperformed by squeezing, right? But again, one could saturate the quantum limit with a cavity. It's not clear at this point how I'm going to build a cavity for something like an electron, right? So we're not going to do that, right? Nor is it clear how to do squeezing operations on these things. Instead, we're going to do what's called a multi-pass measurement. The distinction we're drawing here is that for a multi-pass measurement, I interact my probe with the sample a fixed integer number of times, as opposed to in the cavity case where there's some probability that my light leaks out on every round trip. So this doesn't let me hit the absolute quantum limit per dose, right? But I can still get to about 2/3 of that quantum limit if I set the number of interactions correctly, dependent on the loss that I get from interacting with the sample, right? And so just as in the Raman work I told you about, right, we relied on the long longitudinal coherence of the beam to basically build up the information here. You might worry that that might limit our measurement with these electrons. That's why we can't build an electron cavity, of course. But we verified in the optical domain, right, that even if you use a source with no longitudinal coherence, like an LED, right, you can still get this signal to noise enhancement by going through your sample an integer number of times. So this is just imaging cells in flow we were looking at just with optical absorption imaging, right? But we want to do this with electrons. So how do we do that? We have a custom electron column. This was the design for this was first published back in 2019. The instrument got to Stanford just a few months ago. So one of many long wait times we've heard about today, right? But the way the measurement works is I have an electron source that I trigger within a laser pulse. That electron comes out and it gets accelerated to about 10 keV. And what we're going to use to do this reimaging condition is what we call a gated electron mirror. So typically this mirror is biased up to a potential above the electron energy. So the electron is classically not allowed to go through that region. Now, when it gets close to that mirror, what we're going to do is pulse the voltage down just enough that the electron is allowed to go through. The mirror acts like a strong lens. So we're here now, right? And then it'll get focused onto my sample, which will propagate through. And this optical setup is just mirrored on the bottom here so that I go back through many, many times. So I let my electron bounce around for a while, right, until I'm done with it. And then I know when it's going to approach either the top or the bottom where I want it to come out. And so what I'm going to do is send in another pulse, drop that voltage, and detect. So let's put this just a little bit more in cavity language. Depending on the setting of the mirrors, I have an electron that has a round trip time of about 12 to 20 nanoseconds. So this would be a free spectral range, if you will, of like 50 to 80 megahertz, right? As opposed to caring about something like the waste, right? What's important is the field of view that I get on my sample, right? So that's about 1.5 microns or so. And when I do an electron measurement, we're going to get magnifications on the order of a few thousand. So that puts this field of view right onto the size of your camera chip and will give us few nanometer imaging resolution, which would be inaccessible in optical microscopy, of course, without something like a more complicated super resolution technique. Now, let me tell you about loss in these systems, right? But to understand this a little bit more, you should have a sense of what an electron lens looks like. Right? These are basically hunks of metal that I can bias up to high voltages, at least in the electrostatic case. There's no magnetostatic lenses here, with a very small borehole through the middle of the metal that the electron beam propagates through. And so what that means is that the electron, as it propagates, actually sees no optical material or no material of any kind. It's purely controlled by fields. And so my optics have essentially no loss. What this means is that in the absence of any sample in the column, right, I basically have lossless optics, except of course I'm going to be limited eventually by the buildup of aberrations on the beam as it goes back and forth many times, potentially stray fields, ripples in my power supply, things of that nature. So we think we'll be able to trap electrons for hopefully quite a while in this setup, right? But what happens if we do put a sample in there? Then we need to start thinking about material-based loss processes. So the loss rate for a 10 keV, which is the energy of this instrument interacting with carbon, is about 10%, right? So that would be a finesse of about 60. And rather than talking about a cooperativity, what we're going to call this parameter zeta is the information coefficient. So this relates the amount of elastic scattering I get to the amount of inelastic or damage events. And this is about 0.2 for these 10 keV electrons interacting with carbon, right? But it's about this value 0.1 to 0.25 or so over the range of electron energies from below a keV over hundreds of kilo electron volts, right? So this is pretty low, right? It means on average, I do damage 5 times to my sample before I get any information, right? But what doing this multipass protocol does, right, is it basically increases the amount of elastic events I get per loss event in the sample. So I get an enhancement that goes like m, the number of round trips, times the transmission to the m, right? And so if you do 10 round trips at 10 kV, this factor is about 3.5 or so. So that would boost the amount of information from 0.2 to 0.7. But if you can do more round trips, or if you can go to higher electron energies, it's possible to enter a regime in which this parameter gets over 1, right? And so then basically the amount of information you can extract from your sample is outpacing the amount of damage that you can do. And this is a totally different regime for electron microscopy, of course, but it's one in which you might actually be able to do imaging of single biomolecules before you completely blow them up. So this is what the instrument looks like in the lab. It's a totally custom column. The gun is built by DeLong Instruments, which is a Czech company. The rest of the column and the electron lens stack by Kimball Physics. It's got way more elements than I'm looking forward to controlling. One thing you should see is that these big cans here are the high-speed transmission lines that basically do this pulsing. So the inside of these are biased up to about 10 kV, and then we'll generate these about 100-volt pulses on top of that to bring the electrons in and out of this re-imaging device. So we have an electron beam at the bottom of the column. It looks like this, nice and round these days, right? But the most recent results just this week show that we're now detecting single laser-triggered electrons at the bottom of the cavity. So what you're looking at here is just a whole bunch of time traces from the output of a PMT. And in this trace on the top, we're coming in with many photons per laser pulse. And so you get this set of, there's 10 of them here, equally spaced peaks that are much taller than all the background dark counts. Right? And so what this means is that when I use this high of a laser power, I'm basically in an electron-rich and also an electron photon-rich operating regime. Now the question is, are we going to be able to count individual events, or are we going to be limited by the number of photons we can collect? And so it won't be clear how many electrons we're getting out per pulse. So if I turn down my laser power and keep turning it down, I essentially get into a regime in which I know that I'm electron starved. So the number of electrons coming out of the tip will be Poisson distributed. Most of the electron events will have 0 electrons in them, but I'll get some with 1 and some with 2, etc. And if I'm also photon starved, right, what I will essentially see is a Poisson distribution that goes continuously to 0, right, because the number of photons per electron is also very low. But that's not what we see, right? So I can basically gate on where I know these electron pulses will arrive in time. You can see that sometimes I get an event. I get an event at the same time there, in this trace, a little one in that trace. I don't get an event here. I don't get an event here, etc. Basically make a histogram of these things. And what we see is as we go to lower and lower laser powers is essentially this dip between 0 signal and kind of the Poisson distribution of the photons. And so this dip is characteristic of the fact that my Poissonian statistics of the photon field are actually not the limiting factor of the measurement here, right? This big peak at 0 indicates that we're just electron starved and we can count individual events. Now, the resolution here between single electron events and 2 electron events and 3 electron events is not great yet. We think that the PMT signal for a single electron event is on the order of about 10 millivolts, right? But if we can go ahead and collect more photons with a more optimized setup, we'll be able to better distinguish these and really start to do particle counting or enforce the configuration in which we inject a single electron into the re-imaging cavity to do these experiments. So again, this is just what the microscope looks like in the lab. Worked with a really great team here at Stanford for both of these projects. We also have collaborators in structural biology for the cell growth, for the Raman imaging experiments, Sam Sozaha and Soichi Wakatsuki. For the electron project, we collaborate with industry, Marion Mankos and Tom Tyree. And this work was funded by the Department of Energy and the Gordon and Betty Moore Foundation. So I'd be happy to take your questions.
Great talk, Josh. My question is from the first half. I didn't catch whether you're modulating the pump or the Stokes, whichever one you're modulating, you're detecting the other. With the one that you're modulating, for maximizing your signal in SRS, you typically want to hit it pretty hard. if you change the power of the modulated beam, do you get that 8 or 9 dB of enhancement kind of like across a range, or is it a function of the power of that beam?
Yeah, so this is a good question. Basically, the signal you should expect for SRS is linearly dependent on the pump power, right? And as we turn up the pump power, we get these linear increases in the Raman signal. Yeah. And so the enhancement you get is independent of the pump power. And the comparisons we're doing are always normalized to the dose that you would apply for an equivalent single pass measurement. So this is a 9 dB enhancement, not relative to like the light that gets into the cavity when I don't have it locked. It's relative to applying the same optical field to the sample in a single pass configuration. The way you should think about that is when I do that but don't have the cavity, I send this huge pulse of light onto my photodiode and I get much higher shot noise in particular. Yeah.
Thank you. I also have a question about your first part. So when you do the spectral focusing, so do you need to worry about any like cap dispersion caused by these chirp pulse?
Yeah, that's a good question. So if our bandwidths were super, super broad, right? The cavity modes, yeah. Let me step back. So if I have dispersion in my optical cavity for transmission peaks separated by many free spectral ranges from the center, will be shifted from being perfectly spaced due to the additional round trip time or the additional phase acquired in a round trip. And so if my laser bandwidth was sufficiently high such that I had transmission peaks that extended out to those regimes, right, I would get those lines would not be perfectly resonant with the cavity. And so I would have additional problems from dispersion. Basically, the laser bandwidths are not big enough for this to be a problem, and the finesse is sufficiently low that it doesn't affect the measurement. It doesn't change the transmission. But what we do see is like, yeah, you can totally detect the Raman-induced phase that shifts the lines that we do have. If that makes sense.
Yeah, a nice work. So if I want to get the Raman spectrum, so with the cavity enhancement, what kind of scanning range that's realistic to do?
So the range that we scan over is limited by the pulse bandwidth. And then by your signal to noise, basically. So we're looking at bandwidths of a few hundred wave number. We can collect those spectra in a second easily. Well, it depends on the concentration, of course, but for the data I showed, you can easily do it in less than a second. It's just limited by the speed of your delay stage.
Thanks for the nice talk. For your electron cavity, is there a trade-off regarding spatial resolution and number of round trips? So will you lose your focusing when you have more round trips? And will that kind of limit how well you can image or sense localized features?
In the short term, yes. Now, especially because the instrument is designed for like a lower resolution than is achievable with kind of the commercial microscopes that you get now, right? What you'll eventually be limited by as you go through many, many times is the buildup of aberrations on the electron beam. Now, there's some configurations in which you can correct these things using the mirrors and other electron optics. I think it's kind of an open question how deleterious going back and forth many, many times will be. It depends on the quality of your optics. It also depends on how well you can keep the electron separated from the pulsing on the mirror. What you're worried about is fringing, like ringing in the mirror or something like that. As long as you can pulse much faster than the electron is going to be nearby, you'll be okay.
I had a question of my own on the slides about your quantum limits. Why is it the ultimate quantum limit with a cavity, even in the limit all the way to the end right side of the plot, is better than the noon state?
Because sorry, let me make sure I understand.
Yeah, like right here,
right here.
Noon state.
So for noon states, basically the plot doesn't go to. I have to think about that more. I guess you can choose. I'm not sure why I'd have to think about this more, why at transmissivity 1, right, a noon state measurement is not optimal. But the number of photons that I put in my noon state n is going to be a function of loss. Like for an optimal noon state measurement, I pick a number of n based on the expected loss, right? So I would guess that for in the limit, right, as the transmissivity is going to 1, right, like even still the optimal number of measurements can be outperformed with a squeeze state. So I'm performing worse than the quantum limit.
And a cavity can optimize, like if you just have a cavity with a single photon or like a coherent state of light going into that, that hits your optimal quantum plus cavity limit at the top.
Well, when you think about going to transmissions of 1, right, basically everything can be like infinitely good, if that makes sense, because there's like no penalty to going around more and more and more. So the number of passes that you need to do will diverge, right? And so it becomes impossible to implement. That makes sense. Cool.
Very unitarity limit on that?
Say more?
Well, as the transmission goes to 1, the phase shift per pass goes to 0, right?
From Kramer's, in the limit that the phase shift is totally dependent on this transmission parameter that we're talking about. In the Raman case, right, our transmissive losses are actually dominated by the objectives in the sample itself, as opposed to the resonance that we're probing.
Got it.
But yeah, and similarly in the electron case. So.
Great. If there's no further questions, let's thank Josh one more time.